Hello, everyone, and welcome to this audio podcast. This audio podcast as part of a Phantom edition. This audio podcast as part of a Vertex Roulet. This audio podcast as part of a Lobostics Machina. This audio podcast as part of a Cyclone bonus 2, and this audio podcast as part of a Storm Lobostics production. In this, as part of this, as part of this this audio podcast, this Cyclone bonus 2 audio podcast, this audio podcast will look at a research poster presentation that was put forward for UKKW 2023, looking at observing chemokine and chemokine receptor expression in acute allograft rejection, or AAR, and looking at this research poster presentation, or where this works in this research poster presentation, looks at a retrospective on human transplant immunobiology and laboratory practice. So to begin with, some intro. So renal transplantation is a preferred modality for recipients with end-stage renal disease, or ESRD, who make a sort of good surgical who are good who are good who make who are sort of who are good surgical risks and able to comply with chronic immunosuppression therapies, or IST. Clinical transplantation has advanced significantly, with most transplant institutions transplant institutions reporting 1-year renal allograft prognosis rates of better than 80%. Nevertheless, rejection and progressive loss of allografts over time continue. Over the last decade or so, many reports of chemokine or chemokine receptor expression in context of clinical or experimental allograft rejection have been highlighted. However, there are often numerous ligands for a given chemokine receptor, and research looking at specific chemokine receptors has been increasingly crucial. Now looking at sort of the basis in regard to acute allograft rejection, or AAR. So acute cellular rejection is mediated by lymphocytes which have become alloactive against donor transplantation antigens. In vivo stimulation of alloreactive T cells takes place primarily in the peripheral lymphoid tissues of the recipient, although intragraft sensitization may occur. The strongest stimulus is provided by donor dendritic cells, also referred to as passenger leukocytes, in the allograft, which will enter circulation and end up on the lymphoid tissues of recipient. These the dendritic cells function as antigen presenting cells, or APCs, and they provide a strong stimulus for human leukocyte antigen HLA class 2 reactive CD4 cells, which can stimulate the growth and differentiation of HLA class 1 reactive CD8 cytolytic or cytotoxic lymphocytes. Alloreactive lymphocytes enter the circulation, and they will react with allograft vascular endothelium and the primary target of the initial stage of cellular rejection. Now looking at the sort of underpinnings in regard to chemokine and chemokine receptors and leukocyte trafficking in acute allograft rejection before describing the aims and objectives in around this research poster presentation. So in renal transplantation, the occurrence of one or more episodes of acute allograft rejections or rejection AAR is a major determinant of allograft survival prognosis. Most episodes of AAR are caused by cell-mediated processes and require the infiltration of alloactive T cells into the engrafted organ. Over 40 inducible chemokines and 20 inducible chemokine receptors have been identified to date. In vitro investigations of responses of activated T cells to inflammatory chemokines suggest substantial redundancy in this system, with multiple chemokines produced at tissue sites binding to multi-receptors on circulating leukocytes. Chemokines binding to receptors on leukocytes mediate sort of the behind-the-scenes plays between antigen presenting cells or APCs and host T cells in lymphoid tissues. And the actual here and now to propagate an immune response that acutely destroys a graft and seems apparent as sort of smoldering inflammatory responses seen clinically as chronic rejection. Now looking at some of the aims and objectives. So this is as part of this research poster presentation, and as part of this research poster presentation, and there has been drafted manuscript in regards to this works in regard to sort of an update on this works um, in on this works. The aims and objectives the aims and objectives was where this research poster presentation sought to provide a retrospective overview of observing chemokine and chemokine receptor expression in acute allograft rejection or AAR in context of human renal transplantation and transplant immunobiology. Now looking at transplant tolerance. So, of approximately 18 chemokine receptors, at least 3 appear to play important roles in rejection, and where in amongst laboratory investigations and amongst laboratory investigations, there have been there have been works in regard to experimental investigations linked to CCR1, CXCR3, and CCR5. In experimental research or investigations, the chemokine receptor antagonists are effective are more effective, when used with low dose calcineurin inhibitors or CNIs than when administered as monotherapy or treatments alone. Developmental investigations are in place or have been in place for antagonists of CXCR3 and CCR1. It have been transparent from retrospective investigation that blockade of chemokine receptors have become essential topics and an addition to the therapeutic prevention of acute allograft rejection or AAR. The combination of clinical expression of certain chemokine receptors during rejection with compelling evidence from rodent laboratory investigations using knockout murine and or neutralizing monoclonal antibodies has led to the consideration of therapeutics and targeting of CXCR3 and CCR5 To this end, pharmaceutics have been developing small molecule antagonists and humanized monoclonal antibodies that have been tested or investigated in preclinical investigations, either alone or in conjunction with IST. The first peer-reviewed data on such a work in non-human primate allograft recipient have been available following research since in around 2003, and works by and following on from Hancock and team in around 2003. Now looking at a little bit in regard to renal transplant immunobiology and antigen presentation. So organ transplantation has a long history, beginning with skin autografting in India during the 6th century BCE. The procedure entered Western medicine during the early Renaissance, and is described in a text on the restoration of nose, lips, and ears by Gaspar and Tagliacozzi between 1545 and 1599. In 1906, Matteo Jabolet carried out the first first attempts at human kidney or renal transplantation. Jabolet used pig or swine and goat kidneys, anastomosed or joined to blood vessels of the arm of recipients with chronic renal failure, which functioned for approximately 1 hour. In 1911, Hammond and Sutton of Philadelphia performed the first human-to-human renal transplant or kidney transplant with transient success. As part of this research poster presentation, and an extension, if you like, an extension, a sort of a sort of sorry, a sort of an addition, an additional sort of overview, if you like, of the chemokine receptor to a previous audio podcast in this phantom edition. Where a previous one previous audio podcast in this edition looked at transplant neuroimmunobiology, the immune system, transplant rejection, and neuroendocrinology, a retrospective. And wherein the audio podcast there was an overview in regard to the biology of chemokine and receptors. As part of this, or in this audio podcast, a look at chemokine receptor family, the chemokine receptor family, and looking at this from a sort of more detailed basis. So in regard to the chemokine receptor family, and works originally published in works by originally works are described in regard to the chemokine receptor family by Rossi and Zlotnick in around 2000. And so in Rossi and Zlotnick's work in around chemokine receptor family, there are varying schematics, and so And so a sort of a description, sort of an overview description of an additional schematic, additional sort of look at chemokine receptor family in as part of this research poster presentation. So several points have emerged. From these, from in vivo based mechanistic investigations, first targeting of a single chemokine is typically ineffective in prolonging allografts prognoses. An example of this general principle is a lack of efficacy in targeting a macrophage inflammatory protein 1 alpha arantes, whereas targeting of their receptors, for example CCR1 and CCR5, is of therapeutic significance. Second, chemokine receptors differ in importance as targets in allo responses. Thus, chemokine receptors such as CCR1 and CCR2 primarily promote macrophage recruitment to an allograft, so such that the targeting in knockout mice or murine in amongst murine investigations has only a modest effect on allograft prognoses. And third, the effect of containment IST or immunosuppression can modulate some of the rather black and white outcomes of chemokine receptor targeting in other untreated mice or murine investigations. In the case of chemokine receptors with the most impressive experimental data or investigations on its role in allograft rejection, expression of CXCR3 and its ligands is closely associated with acute allograft rejection clinically. The various stimulatory and inhibitory pathways involved in these activation cascades are providing or have provided insight into the design of potential pharmacotherapy or pharmacological treatments. In the schematic, there is a look at varying chemokine receptors, and then where varying chemokine receptors varying chemokine receptors have the applicable ligands ligands and sort of ligands and sort of chemotactic link between the chemokine receptors and chemokines and the respected uh, and the respected sort of link where the where mechan where in vivo or investigations have identified sort of the mechanistics. So in where CXCR3 and where, for example, CXCR3 is concerned, there's chemotac there are there's in amongst investigations in amongst laboratory investigations or investigations, CXCR3 is chemotactic is chemotactic for IP10, MIG, and ITAC. In regard to CCR5, for example, it is chemotactic for MIP1 alpha, MIP1 beta, RANTES, and MCP2. In regard to CCR7, it is it has been chemotactic for CCR7, chemotactic for MIP MIP 3 beta and SLC. So in amongst the original works by Rossi and Zlotnick in around 2000. Rossi and Zlotnick in around 2000. The authors sort of delve into looking at again the chemokine receptor family more in more detail. So now looking at as part of this research poster presentation in regard to transplant tolerance. So of approximately 18 chemokine receptors, at least 3 appear to play important roles in rejection in experimental investigations: CCR1, CXCR3, and CCR5. In experimental research or investigatory models, the chemokine receptor antagonists are more effective when used with low-dose CNIs or calcineurin inhibitors when administered as monotherapy. And as monotherapy. As monotherapy, and the combination of clinical expression of certain chemokine receptors during a rejection where there's been compelling evidence from laboratory investigation where rodent models investigations and using knockout murine or mice and or neutralizing monoclonal antibodies have led to the consideration of therapeutic targeting again in around CXCR3 and CCR5. And so now looking at sort of the sort of conclusions around this research poster presentations. So in a clinical representation original works by Hancock and team in around 2000 in around between 2000 and 2003. T cells within, for example, bronchial T cells within a bronchial alveolar lavage or BAL of lung transplanted recipients undergoing rejection episodes. Evidence of CXCR3 positive was exhibited with a strong in vitro chemotactic response to IP10. Graft infiltrating macrophages expressed IP10, and alveolar macrophages also expressed and secreted IP10 at levels capable of inducing chemotaxis of CXCR3 positive T cell chemotaxis of a CXCR3 positive T cell line. IP10 production was upregulated by interferon gamma, while high levels of CXCR3 ligands were verified within BAL fluids of recipients with rejection episodes. Overall, our research, our investigations retrospectively inform that there are direct effects of chemokines on recruitment, proliferation, and activation of of vascular smooth muscle and other non-hemopoietic cells. In addition to mediation of leukocyte recruitment, induction of CXCR3 and IP10 messenger RNA or mRNA has been associated with acute allograft rejection or AAR, aligned to human renal transplantation and transplant immunobiology. In this research poster presentation, there are a few acknowledgements and a few contemplations in regarding to those who have lost have unfortunately lost their lives to particular and specific forms of kidney disease that required transplantation, and so that draws this audio podcast to a close. The research poster presentations are formally available through the Renal Patient Support Group, the RPSG platform, through the RPSG platform, where the research poster presentations are available online, and as part of this audio podcast. As part of this audio podcast, this Phantom Edition and this Cyclone bonus 2, there will be a list of applicable references or citations that are linked to this research poster presentation, where listeners are more than welcome to check out the works on similar and around this works and similar works. So that draws this audio podcast to a close. This audio podcast as part of a Phantom Edition. This audio podcast as part of a Vertex Roulet. This audio podcast as part of a Lobostics Macula. This audio podcast as part of this audio podcast as part of a Storm Lobostics production, and this audio podcast again as part of a Cyclone bonus 2.